Have you ever stopped to think about how every single part of a plant, like even down to the tiny cells, has been shaped by evolution? It's not just about looking pretty, it's about function, surviving and thriving over millions of years. And for anyone who loves to understand how things work, well, plants are a gold mine of intricate design.
It's easy to take plants for granted. They just kind of exist in the background, right? But when you really dive into their structure, it's mind-blowing how complex and organized they are. Not just sitting there passively, they're actively responding and adapting to their environment.
Totally. And that's what we're going to unpack in this deep dive. We're lucky enough to have an expert here who's brought along a chapter that's basically the blueprint for vascular plants, how they're built, how they grow, and how they develop from a single cell into these intricate organisms that are essential for life on land as we know it. We're aiming to extract the key concepts, the research highlights, and most importantly, why this all matters to you without getting bogged down in technical terms.
Understanding plant anatomy and development is really the bedrock of so much. It impacts everything from agriculture and food production to the health of entire ecosystems. So by exploring this chapter, you're gaining a deeper understanding of some really fundamental processes in the natural world.
Think of this as your shortcut to plant knowledge. We're going to break down how plants are organized in layers, starting with cells that group together to form tissues, and then those tissues forming the organs we all recognize: roots, stems, and leaves. We'll look at how they grow, both taller and wider, and we'll uncover the mechanisms that give them their shape and allow their cells to specialize for different tasks. So, are you ready to get into it?
Absolutely. Let's start with the concept of hierarchical organization in vascular plants, which the chapter lays out in concept 35.1. Imagine it like a company with different departments all working together. The cells are the basic units, the employees, if you will. They team up to form tissues, which are like departments with specific roles.
Hmm.
And then these tissues come together to form the organs, the bigger functional units like roots, stems, and leaves.
And this layered structure isn't just random, right? Figure 35.1 really drives home the point that natural selection has favored this organization because it works. Every cell type, every tissue, every organ has a purpose that contributes to the plant's survival and ability to reproduce. Like even something as simple as a root hair. It's not just there for looks. It's a brilliant adaptation for absorbing water and nutrients from the soil, which is crucial for plants living on land.
Exactly. And when we think about the three main plant organs, roots, stems, and leaves, we have to remember the evolutionary context. Plants had to solve some major challenges when they moved from water to land. They needed ways to anchor themselves, to obtain resources from both the soil below and the air above, and to move those resources efficiently throughout their bodies.
Which brings us to the root system, the plant's anchor and its lifeline for absorbing water and minerals. The chapter explains that the primary root, which is the first one to emerge, branches out into lateral roots, forming this intricate network. Some plants go for a taproot system, with one strong central root and smaller lateral roots branching off. Think of a dandelion. That deep taproot helps it anchor firmly, especially in drier soils. Other plants develop a fibrous root system, which is more of a dense mat of slender roots closer to the surface. This is great for preventing soil erosion because it holds the topsoil together really well, like the chapter mentions in connection with figure 30.16.
It's interesting to think about the trade-offs, right? A strong taproot takes more energy to grow deep, but it provides excellent anchorage for larger plants. A fibrous root system is efficient for absorbing nutrients near the surface, but it might not anchor a giant tree as effectively. We also see adventitious roots, which can sprout from stems or leaves, giving plants flexibility for things like propagation. And we can't forget about root hairs, those incredible extensions of epidermal cells. Imagine the surface area they add. In some plants, it's like having several football fields worth of absorptive surface area, which is crucial for maximizing nutrient uptake from the soil.
It's amazing how they constantly reach out, exploring for resources. And then there are those specialized root adaptations shown in figure 35.4. Buttress roots, for instance, those wide above-ground roots you see in some tropical trees, help stabilize them in shallow soils. Strangling aerial roots, like those of strangler figs, can actually wrap around and eventually kill host trees talk about aggressive. Prop roots, like in a corn plant, provide additional support. Pneumatophores, also known as air roots, which you often see in mangroves, allow them to get oxygen, even when they're growing in waterlogged soils. And storage roots, like carrots and sweet potatoes, are basically nutrient storehouses packed with carbohydrates and other goodies.
It really shows how natural selection has shaped roots to meet the demands of all kinds of environments. Now, let's move above ground to the shoot system, which is made up of stems and leaves. Stems are the structural backbone, providing support for the leaves so they can soak up sunlight, which is the ultimate energy source for the whole plant. Stems also lift up reproductive structures like flowers, which helps with pollen and seed dispersal. Stems have nodes where leaves attach, internodes, the sections between nodes, and buds. Apical buds at the tip allow for upward growth, and axillary buds, tucked in the angles where leaves meet the stem, can develop into new branches or flowers.
And stems, like roots, can be modified in all sorts of fascinating ways, as figure 35.5 shows. Rhizomes are underground stems that grow horizontally, like ginger, storing food and allowing the plant to spread asexually. Stolons are stems that run along the ground, like strawberry runners, also for asexual reproduction. And tubers, like potatoes, are basically swollen tips of underground stems packed with starch. The figure caption cleverly asks which of these structures has nodes, reminding us that nodes are a key feature of stems that helps us distinguish them from roots.
Exactly. Nodes are the giveaway. Now let's talk about leaves, those solar panels of the plant world. They're the primary sites of photosynthesis, capturing light energy and converting it into sugars. They also exchange gases with the atmosphere and help regulate the plant's temperature. The basic structure of a leaf includes the blade, which is the wide, flat part, and the petiole, which is the stalk that attaches the blade to the stem. Some monocots, though, like grasses, don't have a petiole. Instead, the base of the leaf forms a sheath that wraps around the stem.
Another key difference between monocots and eudicots is the pattern of veins in their leaves. Monocots typically have parallel veins, like train tracks running the length of the leaf, while eudicots have a branching network of veins, often radiating out from a central midrib. Figure 35.6 shows this distinction and also illustrates the difference between simple leaves, which have a single undivided blade, and compound leaves, which have a blade divided into multiple leaflets. The chapter even suggests that this compound structure might help contain the spread of pathogens or damage, since each leaflet is somewhat isolated.
It's like having built-in compartments. And leaves, just like roots and stems, can be highly modified for specific functions. Figure 35.7 shows some cool examples. Tendrils help plants climb by wrapping around supports. Storage leaves and bulbs store food reserves. Spines and cacti protect against herbivores and reduce water loss. And some reproductive leaves can even sprout new plantlets along their margins.
Now let's zoom in a bit and talk about the three main tissue systems that run throughout the entire plant: dermal, vascular, and ground tissue, as illustrated in Concept 35.1 and Figure 35.8. They're not isolated compartments. They're all interconnected, forming a continuous network.
Think of dermal tissue as the plant's outer skin, providing protection. In non-woody plants, the dermal tissue is the epidermis, which is often coated with a waxy cuticle to minimize water loss. In woody plants, the epidermis is replaced by periderm as the plant ages and increases in girth. The epidermis is also home to specialized cells like root hairs, guard cells that regulate gas exchange through stomata, and trichomes, those hair-like structures that can have various roles. As Figure 35.9 shows, trichomes can help reduce water loss in desert plants or even deter insects with their spiky or glandular surfaces.
Next, we have the vascular tissue, which acts like the plant's circulatory system, transporting water, minerals, and sugars throughout the plant body. Xylem carries water and dissolved minerals upwards from the roots to the rest of the plant, while phloem transports sugars, the products of photosynthesis, from the leaves to where they're needed for growth or storage. The term stele refers to the central vascular cylinder in roots and stems. In stems and leaves, the vascular tissue is often organized into vascular bundles, which are distinct strands containing both xylem and phloem. It's a two-way street. Xylem moving things up, phloem moving things down, keeping everything connected.
And lastly, there's the ground tissue, which fills in the spaces between the dermal and vascular tissues. It's kind of like the plant's general-purpose tissue. Ground tissue includes the pith, which is internal to the vascular tissue, and the cortex, which is external. It plays a vital role in storage, photosynthesis, support, and short-distance transport.
Now, to really understand how these tissues work, we need to look at the cells that make them up. The chapter showcases the diversity of plant cells in Figure 35.10, emphasizing cell differentiation, the process by which cells with the same genetic blueprint become specialized for particular tasks. It's like how in a company, everyone has the same employee handbook, but people have different roles and responsibilities depending on their department.
First up are parenchyma cells. These are the most common type, with relatively thin walls and a large central vacuole. They're the workhorses, carrying out essential metabolic functions like photosynthesis, food storage, and even wound healing. Importantly, they retain the ability to divide and differentiate, which allows plants to adapt and repair damage.
Then there are collenchyma cells. They have thicker walls than parenchyma cells, providing flexible support, especially in young, growing parts of the plant. Think of the strings in celery. That's collenchyma.
And then we have sclerenchyma cells, the tough guys. They have thick, rigid walls that are often strengthened with lignin, making them incredibly strong and supportive. Many sclerenchyma cells are actually dead at maturity, their rigid walls forming a kind of internal skeleton. We have two main types. Scleroids, which are shorter and irregularly shaped. Think of the hardness of nutshells and the gritty texture of pears and fibers, which are long and slender, often occurring in bundles. They're what gives strength to things like rope and linen.
Next, we have the water-conducting cells of the xylem, tracheids, which are found in all vascular plants, and vessel elements, which are more common in flowering plants. Both types are dead and hollow at maturity, forming continuous tubes for water transport. They have pits, which are little pores that allow water to move sideways between cells, and vessel elements also have perforation plates, which are basically open ends that make water flow even more efficient. Figure 6.27 provides a closer look at these structures.
Finally, there are the sugar-conducting cells of the phloem. In seedless vascular plants and gymnosperms, these are sieve cells. In flowering plants, we have sieve tubes, which are chains of individual sieve tube elements connected by sieve plates with pores, allowing sugars to flow through. These cells are pretty unique because they're alive at maturity, but they lack many of the usual organelles, like a nucleus, to make way for efficient sugar transport. Companion cells connected to sieve tube elements via plasmodesmata, tiny channels that connect plant cells, provide metabolic support. Again, figure 6.27 illustrates this.
So understanding this hierarchy from cells to tissues to organs really gives us a glimpse into how a plant is built and how all the different parts are connected and work together. Concept check 35.1 asks how vascular tissue enables leaves and roots to function as a unit. Basically, it's all about transport. The xylem carries water and minerals up from the roots to the leaves where they're used for photosynthesis. Then the phloem carries the sugars produced by photosynthesis down from the leaves to the roots for energy and storage.
Right. It's a beautiful cycle. The what-if question about humans being photosynthetic is kind of fun to think about. We need a huge surface area of leaves to capture enough sunlight to power our bodies. And the mention of central vacuoles and cell walls from back in concepts 6.4 and 6.7 reminds us that these features are essential for plant cell structure. The central vacuole helps maintain turgor pressure, which keeps plants upright and rigid, and the cell wall defines the cell shape and provides support.
Now let's shift gears and talk about how plants actually grow, specifically how they increase in size. Concept 35.2 introduces the idea of indeterminate growth, meaning plants can keep growing throughout their lives thanks to special tissues called meristems. This is a big difference for most animals, which have determinate growth and stop growing once they reach a certain size. Imagine a tree that keeps getting taller and wider year after year. That's indeterminate growth.
The chapter highlights two main types of meristems. Apical meristems are located at the tips of roots and shoots, and they're responsible for primary growth, which is all about increasing the length of the plant. This allows roots to delve deeper into the soil and shoots to reach higher for sunlight. Lateral meristems, which include the vascular cambium and the court cambium, are responsible for secondary growth, increasing the girth or thickness of woody plants. So primary growth is about getting taller, and secondary growth is about getting wider.
Figure 35.11 gives a great visual of this, showing how apical meristems drive vertical growth, while lateral meristems add layers for radial growth. Meristems contain initial cells, which are like stem cells in animals. They can divide indefinitely, producing more meristematic cells, and also cells that will differentiate into specialized tissues.
The winter twig example in Figure 35.12 illustrates the relationship between primary and secondary growth really well. You can see the dormant apical bud ready to burst into growth next spring. Below it are leaf scars, showing where leaves were attached, and axillary buds, which have the potential to form new branches. Bud scars show where the protective bud scales were in previous years. It's like a record of the twig's growth history.
It's like looking at tree rings, but on a smaller scale. Concept check 35.2 raises some interesting questions, like can primary and secondary growth happen at the same time in the same plant? Absolutely. Think of a young tree. The tips of its branches are extending, primary growth, while its trunk is also thickening, secondary growth.
Another question is, why do roots and stems have indeterminate growth, while leaves generally don't? Well, roots and stems need to keep growing to access resources. Water and nutrients in the soil for roots, sunlight for stems. Leaves, on the other hand, have a more defined lifespan, and their primary function is photosynthesis, which they can do efficiently at a certain size.
Let's dive a little deeper into primary growth, specifically the lengthening of roots and shoots. Figure 35.33 shows us a close-up of a root tip. The root apical meristem, which is the zone of active cell division, is protected by the root cap, a layer of cells that secretes a slimy substance to help the root push through the soil. Behind the apical meristem are three zones: the zone of cell division, where new cells are born; the zone of elongation, where cells grow longer, pushing the root tip forward; and the zone of differentiation, where cells specialize into their mature forms.
And within those zones, the primary meristems are hard at work. The protoderm, the outermost layer, gives rise to the epidermis, which is the protective skin of the root. The ground meristem gives rise to the cortex, the region between the epidermis and the vascular tissue. It's mostly made up of parenchyma cells for storage and transport. The innermost layer of the cortex is the endodermis, which acts as a gatekeeper, controlling what moves into the vascular cylinder. Figure 36.9 goes into more detail about that.
The precambium, the innermost primary meristem, forms the vascular cylinder, or stele, which is the core of xylem and phloem in the root. Figure 35.42 shows how this is organized. In eudicote roots, the xylem forms a solid core that often has a star shape, with the phloem nestled between the arms of the star. In monocote roots, there's usually a central core of parenchyma surrounded by alternating rings of xylem and phloem. Also, the pericycle, which is the outermost layer of cells in the vascular cylinder, is where lateral roots originate. They push out through the cortex and epidermis to form new branches.
Now let's switch to primary growth in shoots. Figure 35.16 shows the shoot apical meristem, a dome-shaped cluster of dividing cells protected by tiny leaves within the apical bud. Leaf primordia, which are basically baby leaves, emerge from the sides of the apical meristem. Shoot elongation mainly happens because the cells in the inner nodes, the spaces between the nodes, lengthen. Just like in roots, the protoderm, ground meristem, and precambium are active in the shoot apical meristem, giving rise to the epidermis, ground tissue, and vascular tissue of the shoot.
Branching in shoots comes from the activation of axillary buds, which are located where leaves meet the stem. Here's a cool concept: apical dominance. The apical bud at the top of the shoot releases hormones that actually inhibit the growth of axillary buds further down the stem. This ensures that the plant prioritizes growing taller, towards the light, rather than branching out too much. But if you remove the apical bud, either by cutting it or if an animal eats it, those axillary buds are released from inhibition and start growing, resulting in a bushier plant. Gardeners use this principle all the time when they prune plants.
Stem anatomy is a little different in eudicots and monocots, as you can see in figure 35.17. Both have an epidermis, ground tissue, and vascular bundles, but the arrangement is distinct. In eudicot stems, the vascular bundles are arranged in a ring, with the xylem on the inside closer to the pith and the phloem on the outside closer to the cortex. In monocot stems, the vascular bundles are scattered throughout the ground tissue, so we don't really see a clear distinction between pith and cortex.
Figure 35.18 shows the anatomy of a leaf. Leaves develop from leaf primordia, produced by the shoot apical meristem. The leaf's epidermis has a waxy cuticle to prevent water loss, and stomata, those tiny pores flanked by guard cells, allow for gas exchange. The ground tissue inside the leaf is called mesophyll. In many eudicots, it's divided into a palisade mesophyll, with tightly packed cells specialized for light capture, and a spongy mesophyll, with air spaces for gas exchange. Veins, which contain the vascular tissue of the leaf, are continuous with the vascular bundles in the stem, bringing water and nutrients in and taking sugars out.
Concept check 35.3 asks us to compare primary growth in roots and shoots. Roots have that protective root cap, and their growth is very linear, focused on pushing forward through the soil. Shoots are protected by the leaves of the apical bud, and their growth is more about elongating the internodes. Branching is also different. Lateral roots emerge internally from the pericycle, while lateral shoots sprout externally from axillary buds.
So we've covered how plants grow longer. Now let's talk about how they grow wider, which is secondary growth, covered in concept 35.4. This is the process that gives us wood and trees. The chapter points out that secondary growth occurs in most gymnosperms and many eudicots, but it's rare in monocots and leaves. Figure 35.9 provides a great overview of secondary growth in a woody stem.
Secondary growth is all thanks to two lateral meristems: the vascular cambium, which produces secondary xylem wood and secondary phloem, and the cork cambium, which forms the outer protective layer called periderm.
The vascular cambium is located between the primary xylem and primary phloem in a woody stem or root. It's a cylinder of actively dividing cells, and as they divide, they add new layers of secondary xylem to the inside and secondary phloem to the outside, increasing the girth of the stem or root. Some vascular cambium cells also produce vascular rays, which are like spokes running radially through the wood, transporting materials laterally and providing storage.
Secondary xylem, or wood, is mostly made up of tracheids and vessel elements, which are those water-conducting cells we talked about earlier, heavily reinforced with lignin for strength. Figure 35.20 shows the annual growth rings that form in trees in temperate climates. Early wood, produced in the spring, has larger cells with thinner walls to maximize water transport during the growing season. Late wood, produced later in the year, has smaller cells with thicker walls for more structural support. These rings are like a historical record, and scientists use them for dendrochronology, studying past climate and environmental conditions.
It's amazing how each ring tells a story. The chapter also distinguishes between heartwood, which is the older non-functional xylem in the center of a tree trunk, often filled with resins and other compounds that make it resistant to decay, and sapwood, the younger functional xylem that actively transports water. Figure 35.23 even shows how a tree can survive with a hollow heartwood as long as the sapwood is intact, carry water up to the leaves.
Secondary phloem, which is produced by the vascular cambium toward the outside of the stem or root, is responsible for sugar transport. However, unlike secondary xylem, which accumulates as wood, older layers of secondary phloem are sloughed off as the stem or root expands.
Now, the cork cambium is the other lateral meristem involved in secondary growth. It develops from the cortex in stems or the pericycle in roots, and it produces cork cells. These cork cells are impregnated with suberin, a waxy substance that makes them impermeable to water and gases. The layers of cork cells form the periderm, the protective outer bark of a woody plant. Bark actually includes all the tissues external to the vascular cambium, so it includes the secondary phloem and all those layers of periderm, as figure 35.2 shows.
Figure 35.9 also shows lenticels, which are small raised areas in the periderm that allow for gas exchange. Even though the periderm is mostly impermeable, the living cells inside the stem or root still need to breathe.
The chapter even touches on the evolution of secondary growth, mentioning research on Arabidopsis that suggests secondary growth might have evolved from modifications of primary growth pathways. Figure 35.24 provides a nice summary diagram showing the different primary and secondary tissues in the woody stem.
Concept check 35.4 has some thought-provoking questions. For instance, if you hammered a nail into a tree trunk 5 feet above the ground, would that nail be at the same height 5 years later? The answer is yes, because trees grow taller from their apical meristems, not by elongating the existing trunk.
Okay, now let's wrap things up by delving into concept 35.5. Growth, morphogenesis, and cell differentiation. The key processes that shape a plant's development. Development is basically the sum of all the changes that occur as a single fertilized egg cell transforms into a whole multicellular organism. In plants, this includes growth, an irreversible increase in size morphogenesis, which is the development of form and the arrangement of tissues and organs, and cell differentiation, where cells become specialized for different tasks. Plant development is influenced by both genes and the environment.
The chapter highlights developmental plasticity, which is a plant's ability to alter its development based on environmental cues. Figure 35.25 shows a great example with Cabamba aquatica. This aquatic plant develops different leaf shapes depending on whether it's submerged in water or floating on the surface. This kind of flexibility is super helpful for plants which can't just pick up and move to a different location if conditions aren't ideal.
The chapter also mentions the importance of model organisms like Arabidopsis thaliana for studying plant development. Arabidopsis is like the lab rat of the plant world. It's small, it has a short life cycle, it produces lots of seeds, and its genome has been fully sequenced. Figure 35.26 shows how researchers are studying natural variation in Arabidopsis to understand how plants have evolved and adapted to different environments, which could lead to improvements in crops.
Growth in plants happens through cell division, which increases the number of cells, and cell expansion, where individual cells get larger. Cell expansion is actually the main driver of plant growth. It's mostly fueled by water uptake into the central vacuole, which is a very efficient way to increase size. Think of how fast bamboo shoots can grow. Figure 35.28 illustrates anisotropic expansion, which is when cells elongate in a specific direction, guided by the orientation of cellulose microfibrils in the cell wall and microtubules in the cytoplasm.
Morphogenesis and pattern formation are all about how a plant develops its overall shape and how its tissues and organs are arranged in a very specific way. We always have the dermal tissue on the outside and the vascular tissue on the inside, right? The chapter talks about two main models for how cells figure out their identity and position during development. Lineage-based mechanisms, where a cell's fate is determined by its ancestry, and position-based mechanisms, where a cell's fate is influenced by its location and signals from neighboring cells. Experiments suggest that position-based mechanisms are particularly important in plants.
This is a bit different from animal development, where lineage often plays a bigger role. The chapter mentions that plants have homologs of the homeotic genes that are crucial for development in animals. For instance, the K-NOD1 gene in maize affects leaf shape, and the Mandas box genes are involved in determining the identity of floral organs. Figure 35.30 shows how the DRABR2 gene controls whether Arabidopsis epidermal cells develop into root hairs based on their position relative to underlying cortical cells.
Ultimately, cell differentiation, the process of cell specializing, is controlled by differential gene expression. Different sets of genes are turned on or off in different cell types, leading to the production of different proteins and ultimately different structures and functions. Cell-to-cell communication is essential for this process, as cells send signals to each other that can influence gene expression.
Plants also go through developmental transitions called phase changes, which are controlled by the shoot apical meristem. One example is the shift from a juvenile vegetative phase to an adult vegetative phase, often marked by changes in leaf shape, like in Muhlenbeckia australis, shown in figure 35.31. It's interesting that even after the shoot apical meristem has transitioned to the adult phase, the nodes and internodes that developed earlier during the juvenile phase retain those juvenile characteristics.
The transition from vegetative growth to flowering is another well-studied phase change. It's triggered by both environmental cues like day length and temperature and internal signals. Once a plant starts flowering, the floral growth is determinate, meaning it has a defined endpoint. The chapter describes the ABC hypothesis of flower formation, a model that explains how three classes of genes, A, B, and C, control the development of the different floral organs: sepals, petals, stamens, and carpels. Each gene class is active in two adjacent whorls of the flower, and the combination of active genes in each whorl determines the identity of the organs that form there.
It's like a floral code. Figures 35.32 and 35.33 illustrate this beautifully. For instance, A genes alone specify sepals, A and B genes together specify petals, B and C genes specify stamens, and C genes alone specify carpels. There's also an inhibitory interaction between A and C genes, so they can't be active in the same whorl. By studying mutants that lack one or more of these gene classes, researchers have confirmed the ABC hypothesis.
Concept check 35.5 wraps things up nicely, asking how cells with the same DNA can have such different structures and functions. It all comes down to differential gene expression. Different sets of genes are active in different cells, leading to the production of different proteins, and therefore different structures and functions. The question about tepals floral structures that look like a blend of sepals and petals suggests that the ADC model might be a bit more complex than we initially thought, with some overlap in gene expression between those two organ types.
The chapter concludes by reminding us that the whole plant is an integrated system, and its structure and function reflect its adaptation to life as a photosynthesizer on land.
So, as we conclude our deep dive, I hope you've gained a new appreciation for the complexity and beauty of plant structure and development. Next time you see a plant, whether it's a towering redwood or a tiny wildflower, take a moment to really look at it. Notice the arrangement of its leaves, the texture of its bark, the way its roots grip the soil. Think about the intricate processes happening inside, from the busy meristems to the specialized cells transporting water, nutrients, and sugars.
It's a whole world of intricate design and adaptation all happening right before our eyes. Here's a final thought for you. Given the remarkable developmental plasticity of plants and their ability to adjust their growth and form in response to their surroundings, what can we learn from them about adapting to the environmental changes happening on our planet? Could understanding plant adaptation hold the key to building more resilient ecosystems and ensuring food security in a changing world? It's something to ponder as you go about your day.